Thank you, Mr. Chairman. I think I don't need the microphone. You can hear me, I suppose. So today, first I would like to thank the organizers, specifically Thibaut and his team, for selecting my talk. So I will speak about biomineralization. So we are working on the chicken eggshell biomineralization for a long time, and we are collaborating with Max Inke's laboratory and Alejandro Rodriguez Navarro, Juan Manuel Garcia Ruiz in Granada in Spain for more than 20 years on these topics. And for this aspect that I will introduce today, we have also worked with Valérie Labat, who is the head of the proteomic core facility in INRA in Tours. So the chicken eggshell is original function is not to be eaten. And for the egg, it's just to be a natural envelope to ensure the physical defense of the egg, and its function is to protect the developing embryo and to ensure that the table eggs remain free of pathogens. But also it's a natural ceramic which is formed at ambient temperature and normal pressure, and consequently it's a good model of biomineralization of membrane support. So today I would like to give you an overview on the current knowledge of the chicken eggshell structure. I will give you some detail on recent advances on the mechanisms of eggshell formations, and I will stress more particularly my talk on the pivotal role of the eggshell matrix proteins. So the eggshell biomineralization occurs in the reproductive tract of the bird, in the uterus, and this is one of the fastest biomineralization systems on the Earth. You have 5 to 6 grams of mineral which are deposited within a 20-hour period. And this chicken eggshell is made of calcium carbonate and calcitic polymorph. And there is an organic matrix, 3.5%. It is mainly proteins and proteoglycans. So on the left, you have a scanning electronic microphotography of the different layers of the chicken eggshell. On the right, you have a cross-polarized light, and you can see that it diffracts in the single way. So it's like all the crystals are oriented in the same direction, and to have some pseudo-single crystal inside the chicken eggshell. So from the internal part to the external part, so close to the egg white, you have first some eggshell membranes. They are composed of organic fibers. And on these organic membranes are deposited some organic cores, which are called some mammillary knobs. And these mammillary knobs are the seeding site where the first calcite crystal will start to grow. Finally, the growing eggshell will give some mammillary layer, and you have a fusion of the adjacent cones to give the compact palisade layer. And when you do not have any fusion of the cones, you have some pores. And these pores are essential to ensure the exchange of gas, which are necessary for the embryonic development. And finally, the most external layers, the outer layer, is an organic cuticle, which is covering the entire surface of the egg. And this is this cuticle which ensures that the egg will remain completely sterile, because no virus, no bacteria can penetrate if it remains intact. So this is the reason why it is forbidden to wash the eggs in Europe, for example. Okay, so the eggshell biomineralization occurs in uterus, and in this uterus there is two main phenomena. The first one is that there is a need to supply the large quantity of minerals necessary for the shell mineralization. So in the lab we have worked a lot to describe a general model of the uterine ion transport during the eggshell calcification. So this is active transport. But I will not give you detail today about that. I will more focus on the biomineralization process, and of course there is some interaction between matrix and minerals. There is 3 main phases of the eggshell formation in the uterine fluid. The uterine fluid is a non-cellular milieu. There is first an initiation stage. There is a slow growing rate of eggshell calcification, 0.1 gram per hour. And it corresponds to the first event of crystallization around the mammillary knobs. There is a linear growth phase and rapid and active growth phase, 0.33 gram of calcium carbonate each hour. And finally, there is an arrest of mineralization 2 hours before the ovipositions. And it corresponds to the deposition of the cuticle layer. So the organic matrix is believed to play a key role in this process, and it determines the texture of the shell and consequently its resulting mechanical properties. So when we speak about the ultrastructure and the mechanical property of a chicken eggshell, we have to think that it depends, of course, of the quantity of material, but it also depends of the interaction between matrix and mineral to control the biomineralization process. So I'm not an expert in physics, I'm a biochemist. But what happened is that the natural calcium carbonate with C-axis, you have some adsorption of organic matrix compounds, some inhibition of some specific phase, some new phases which occurs, and you have these such elongated crystals. So the organic matrix play a role to the orientation and stabilization of calcium carbonate polymorphism and to drive the calcite morphology. So what's happened? At the first time of the mineralization event, you have this elongated crystal that's all oriented in all direction. This is the arrows are in the direction of the C-axis. And you can see that at the following time that some adjacent crystal cannot grow because there is some competitive, physical competitive aspect. So only crystal perpendicular to the surface can develop. And you have a preferred orientation in the external part with the C-axis perpendicular to the surface. So, of course, there was plenty of scientific research and investigation that has been performed to identify the eggshell matrix protein. But the situation was that in 2006 we only knew 11 proteins, and major advances came in the last decade. First, because just after the publication of the human genomic sequence, the second genomic sequence that was published was the chicken genome sequence. So we have now a complete genome-wide non-redundant catalog of a lot of genes, and we have more than 600,000 functional genes. So it's make available the use of omics high-throughput techniques and data mining and bioinformatic tools to identify the egg proteins. And what is the situation today is that we know more than 670 proteins in the eggshells and more than 700 specific transcripts associated to this formation. And of course, only a few number of them can be active on the mineralization. There is a lot of them which are minor, but some of them are abundant and active. And what we want to know now is to determine, to quantify the eggshell matrix protein and their variation according to the shell mineralization process. So we have a new French program, IMPACT, that has been granted by the French National Research Agency. And the aim is to determine the molecular actor with a pivotal role during the mineralization process. So there is 4 in our team that was associated to this program, and University of Granada and University of Ottawa. The aim is to obtain a list of protein candidates involved in particular key point and the essential point of the mineralization process. So we have focused a lot of the first events of the shell mineralization process, because we know that during the initial stages there is some crucial event for the determination of the ultrastructure of the biomineral and to determine the final eggshell mechanical property. First, 5 hours post-ovulation, you have the deposition of nucleosides, and at about 6 hours post-ovulation, the first nucleation event. At 7 hours post-ovulation, there you have some additional crystallization event, and you have some large calcite crystal units which are forming. And finally, we have compared these 3 first events to to a stage when you have an active calcification phases. And we have performed some in situ physical measurement. It was made at the University of Granada. And on the same sample, we have used some high throughput quantitative and statistical analysis of matrix spectrometry. So I'm just give you some result about the in situ physical measurement, and I would like to detail more on the role of ACC in the chicken eggshell formation. So you all know that calcite is a very common material to build highly sophisticated protective mineral structure. So calcium and carbonate can give calcite, but also it's really well demonstrated now that ACC can be a very important transient phase to be more soluble, more reactive, and used as a temporary calcium source. But it has never been demonstrated before in the eggshell, chicken eggshell. There is only one publication from Freeman et al. In 2010. We've made some simulation, some modelization of one chicken eggshell matrix protein of Vocladin 17, and they demonstrated that this protein could potentially be able to transform the amorphous calcium carbonate to some calcite in the shell. So Alejandro Rodriguez Navarro in Granada used some FTIR techniques. I will not give you detail because I'm not able to do that. But what is his conclusion is that there is, at the edge of the mineralization, there is a presence of an amorphous transient phase, which is more important during the initial stages. 95% of the calcite is first on ACC form, and after is transformed in calcite. And this phenomenon still remains important during the active calcification phase. What happened? At the first time point of the shell calcification, you can see some ACC particles which are covering the entire surface of the shell membranes, and they form some massive deposit. And you have here the seeding size, the mammillary knobs. And the second time, you have an interface coupled dissolution precipitation process, so where you have a transformation of ACC into calcite aggregate specifically around the mammillary knobs. Then you have some additional crystallization and event on calcite template, and the final result is that you obtain some large calcite crystal unit depositions. So this is this different event that I have summarized in this representation, and what we wanted to know is to associate this event of calcification with the proteins that can be involved in this process. So we have collected the same sample, and we have identified and quantified the protein at the 4 stages, and we have performed some statistical analysis at the 4 stages. And what we observe, I will not give all the detail, but there is 175 proteins which have some differential abundancy depending on the mineralization stages. 27 are overabundant during the first ACC initial deposition stage. 13 during the transformation into calcite. 47 are overabundant during the crystal unit formations. And 70 are overabundant during the rapid calcification stages. So the question arises very rapidly, what is the function? What is the biological activity of the identified matrix protein? So we have used some literature information, some data mining, some bioinformatic tools, and we could classify this protein in 3 different groups according to their potential function. One important group that I will not detail are the antimicrobial proteins. Why? Because this formation occurs in an external milieu, and the inner part of the shell must remain sterile. So you have permanently a sequestration of an antimicrobial protein which are incorporated in the hard shell index. And after you have some protein that play a direct role on the shell mineralization process, and some proteins that are not directly involved in the shell mineralization, but they have an important role in the regulation of the activity of the protein driving the mineralization. So concerning the protein associated to shell mineralization process, there is first some proteins which are already known to be involved in the biomineralization of shell or to the other biominerals, like the bone, like the molluscan, like the chicken eggshell. There is also a lot of calcium binding proteins which are able to interact with calcium to favor the crystal nucleation. And what we have observed, and this is something very common in biominerals, Frédéric Marin, who will be present tomorrow or Wednesday, I don't remember, have also observed that, is that there is a lot of protein with EF and EGF-like calcium binding domains which are present in the biominerals. Also, we observe some proteoglycan and proteoglycans binding protein. And proteoglycans play also an important role because they have a negative charge to attract the calcium ions. And finally, some eggshell membranes proteins. What's about the protein involved in the regulation of activity of protein driving the mineralization? First, there is some proteins which are involved in the proper folding of the eggshell matrix protein. This is very important to have an appropriate conformation of the proteins to ensure that calcium and minerals interaction can occur and to ensure an appropriate template to the mineralization structure. So there is a lot of proteins in all biominerals. All proteomic studies that has been performed of biominerals demonstrate that there is a lot of molecular chaperone or protein assisting folding. And finally, you have to keep in mind that the shell mineralization occurs in a milieu where there is no direct action, there is no cells. So that means that you need to have some direct action of proteins to inhibit or to activate the molecular actors which are present in the milieu. So they can be the role of molecular chaperone, but also be the role of protease and protease inhibitors. This protease and protease inhibitor can control, have a specific and controlled role during the calcification process by degrading the other proteins driving the mineralization or regulating the processes of protein to make them active on the process. And finally, the mineralization depends on the degree of the protein phosphorylation. It has been clearly demonstrated for the chicken for the osteopontin by Maxime Keys. So we paid also particular attention to kinases and phosphatases. So finally, if we classify these proteins as in the different groups, you can see, for example, at this initial stage that almost half are not defined because we cannot explain their role, but we have also 25% which are antimicrobial and the other 25% which are more associated to biomineralization. So the total is more than this number because some of the proteins can have both roles. It's very often. And this is a result for the other stages. So I do not have time now to detail all these proteins, but as a conclusive remarks, I just want to summarize. 2 minutes, that's perfect. The most important one, which are, I will say, the top 20 of the protein involved at each phase of shell calcification. First, the most abundant one are lysozyme and ovotransferrin. This is some protein that has already been described to be able to modify the morphology of calcium carbonate and specifically the calcite. And they are overabundant, this is the red color, at the initial stages. Secondly, a very important role can be defined for vocladin 17. This is the protein that was simulated, some modelization, computer studies have been performed. And this study indicated that she will be associated to the transformation of ACC into calcite, and ovocladin 17 was overabundant at this stage. We also observe ovalbumin, and there is some lot of paper because ovalbumin was used for in vitro study, and it has been demonstrated that ovalbumin can catch, for example, calcium to perform some amorphous calcium carbonate into the internal part of the molecules, and after it there is a denaturation of the molecules and some calcitic forms that appears. Finally, ovomucoid. Ovomucoid is a protease inhibitor that will control the activity of the other proteins. There is also some additional proteins, I will not give you the detail, that are more associated to the first event, but they are related to the proteoglycan metabolisms, for example. And two other important candidates are adyl-free and albumin. Adyl-3 is a protein which contains 3 EGF calcium binding domains. And in all study that has been performed using transcriptomics or proteomics, it is highly expressed in this model. Ovocladin 116 is a specific Excel matrix proteins, very an important amount associated to the rapid growth phase. Clusterin is a chaperone molecules that will control the process. Cystatin 3 is a protease that can degrade or activate some of the protein. And finally, some additional protein that I will not give you some detail here. Okay, so finally, I would like to invite you to join, specifically the young, to the French Mineralized Tissue Biology Days, which are an excellent opportunity to meet and exchange between researchers from France. And this event this year will occur in Superbès, so it's not so bad. You can join and have very intensive scientific actions and finally complete some ski time. Thank you very much.
Thank you very much.